Hi everyone! After talking about the circulatory and respiratory system in the last chapter, in this chapter we will move on to the urinary system. Our urinary system for mammals is all about regulating balance of water contents, as well as solutes and essential nutrients. And we also use it to get rid of any toxin that is present in our bloodstream. So we will again survey some of the other animals' urinary system, as well as talking about some of the evolutionary adaptations for the method of osmoregulation that we and they have today. So let's get started. All right, so let's start by talking about what is osmoregulation to begin with. So animals must control solute concentration as well as balance water gain and water loss in order to be homeostasis for water content and whatever nutrients we need. This is what we refer to as osmoregulation. So remember, for osmoregulation, it's always two things involved: your solute and your water content. You can't go one without the other because water can only move into our cells by osmosis. There's no other way for water to move into our cells. So, what drives water movement is by solute concentration. So, this is why with osmoregulation, you cannot get away with one or the other. You have to have both. Now, there are relative concentrations of water and solute that must be kept within very narrow limits. We're talking about any living things. There's not, a single animal, living being, that has a very wide range of, osmoregulation limits, where they can have really high osmolarity and then really low osmolarity the other day, unless, they're talking about, adaptations that they have for their diet, etc. But in general, we can, these things have to be kept within very narrow limits. You can't have a broad range. So why that's the case is because for sodium ions and calcium ions, we use those ions that functions for muscular and neural functions. So without these ions being in the right concentration, we're going to be in trouble because both of these have to be in homeostasis as well. So not only water have to be in homeostasis, the amount of specific salt or ion you have in the body is also within a specific limit. If you go over or under on calcium, you can end up with muscle paralysis or overactive muscles, overactive neurons, neurons that is not responsive, etc. So everything in your body, as I've kept mentioning since we started the organ system units, is by homeostasis. You have to have everything in balance. Now, because animals, like I said, lack a pump to transport water across cell membranes, water balance is achieved mainly by osmosis. And remember, I mentioned this before, the rule for osmosis is always water always move toward an area with higher solute concentrations, or something shorter that I always tell my students: water always follows salt. Write that down somewhere. Water always follows salt. Once you have that down, then you never mistake how osmosis works. Now, we won't talk about diffusion in this particular chapter in terms of, what gradient they have, etc. But make sure you kind of remember in the back of your mind that osmosis is different from diffusion, okay? Osmosis is only dealing with movement of water, whereas diffusion, we're talking about movement of solute themselves. So this diffusion means that the solute can diffuse through some sort of membrane and they can equilibrate their concentrations. Whereas osmosis, we're talking about movement of water because the solutes cannot pass through that particular semi-permeable membrane. And only water can do the job of equalizing the two sides concentration. I'll talk more about that in a bit. Don't worry, I'll come back to this concept if you're sort of, confused about osmosis ever since Bio 1406 and you want to, be really able to understand it. Because like I said, you cannot get away with higher up biology classes without coming back to osmosis. You come back to it in anatomy and physiology, you come back to it in microbiology, you come back to it in cell physiology, etc. All upper level biology classes. So all right, so in this particular chapter, we no longer use the tonicity words because remember back from Bio 1406, 1406, we talked about osmolarity in terms of hypertonic, isotonic, and hypotonic. In this particular chapter, the tonicity terms are out the door and not used because animal adaptation to different salt environment is what we're focusing on here. Um, Not what will this red blood cell do in this known um, salt concentration solution. Okay, so that's what tonicity meant. We know we have this bag of certain percent salt concentration, 0.01 or 5%. So what would happen if I dunk this cell into the solution? That was tonicity because we were talking about hypertonic solution, isotonic solution, etc., etc. Now in this chapter, we're talking about and still comparing osmolarity, solute concentration, but it's just we're not comparing known concentration. We would just say this is fresh water, this is salt water, what happens, etc. So that's why in this chapter they want to use the osmotic word versus the tonicity word. All right. So if you remember but the concept is still the same. Okay. If you remember this from Bio 1406, all more power to you. If you don't understand it, listen carefully. Okay. So if cells are hyperosmotic to the environment, then water will move into the cell. Now, what does hyperosmotic mean? Remember, hyperosmotic always means higher solute concentration. There is more stuff inside this particular cell. Okay, so it's illustrated here in this particular picture. So inside the cell, there's a lot of those orange circles. Okay, so that those circles represent the amount of solute you have in the solution. Hyperosmotic means there's more orange circle on your side, which means more salt or solute on your side. So when we're talking about osmosis, we said osmosis is where water will always travel towards the side with higher concentration of solutes, or water will always follow salt. So there's a salt, there's a higher concentration. Water will then rush in to the other side and fill this area with more water. So water will, that's why, will move into the cell as you saw here. Okay. And if, let's say there's a huge osmolarity difference or solute concentration differences, then water will keep rushing in, rushing in, rushing in. If that cell cannot withstand that pressure anymore, that cell can lyse. Okay. And in this case, we're no longer talking about red blood cells or any other cells. We're talking about, animal species, like, or the whole organism. What will happen to them? So most likely they will die if you put an animal in hypoosmotic environments, because then that means there's not enough solutes in the environment. Water come rushing into themselves, and the animal will die from their cells being lysed. All right, so same as isotonic, if cells are isoosmotic to the environment, there is no net water movement. Remember, this doesn't mean water is not moving between either sides. It just means that there's no net movement, which means however much water moves from the left to the right, that's how much will be moving from the right to the left. So there's always equal amount of water flow to both sides, such that there is no net water movement. There's no net water movement to one side or the other. That's what we mean by isoosmotic. And this is where the animal or the cell is happy camper, because there's no movement of water into or out of the cell, is best the way it is. Now, if a cell are hypoosmotic to the environment, then water will move out of the cell. So let's look at this picture right here. So if cells are hypoosmotic, what does that mean? It means that there is lower solute concentration inside the cell. So here's inside the cell, and there's now less orange circles on inside of the cell. So compare to outside of the cell, inside the cell has less circles or solutes. So the fact that it has less solutes and water always follows salt, you go outwards because outside has more salt. So water will flow out of the cell. Okay? So in a cell situations case, usually the cell end up shriveling, or what we in red blood cells we call them cremating. But when the cell shrivels, that means water has moved out, and also the cell is not happy. Animal is not happy; they will not survive. Okay, so for animals to survive in the perfect osmolarity or osmoregulation maintenance, they want to be in isoosmotic as much as possible. Okay, so I will talk about that when we get to different situations of different animals living in different environments. Okay, so make sure you rewind and come back to this section if you're still unclear about what is osmosis and what is hyperosmotic, isoosmotic, and hypoosmotic mean. Remember, these things are always with respective to what I'm saying. Like if the cell is hyperosmotic to the environment, then the cell has more solute to the environment. If I switch it around and say the environment is hyperosmotic to the cell, then the environment has more solute than the cell. So make sure you read it correctly as well. Don't go, oh, hyperosmotic, water move in. Okay. You have to look at what is hyperosmotic to what in order to answer the question. Okay. So make sure you're very clear about that. All right. In addition, the other part of this chapter we said that is excretion. So excretion is something where animals have to get rid of junk or waste stuff inside their cells. In the previous chapter, we talked about the waste with gases situation will be carbon dioxide. For digestive purposes, your animals now have another waste called ammonia. Ammonia is actually a toxic metabolite that is produced by breaking down, or what we call hydrolysis, of nitrogen containing molecules. And those are usually end up being proteins and nucleic acids because they are usually more full, abundant in nitrogen containing molecules than the other ones, than carbohydrates or lipids. So when we are eating proteins, eating things with nucleic acids, of course our body will try to break that down. And when they break those down, you end up producing a waste product that is ammonia. Okay, so the nitrogen part of the molecule that you broke down end up turning itself into ammonia inside the body. So that's why it's a metabolite. It's a waste product from metabolism of your macromolecules, proteins, and nucleic acids. Now, it is a very toxic chemical molecule. So all animals' body have to get rid of this toxic metabolite, and we call that excretion. Something exiting out of your body, and you're getting rid of it. We'll talk about how animals get rid of this molecule, and usually sometimes there's even conversions of changing this ammonia to something less toxic in the body for other animals. All right, so let's start by talking about osmolarity again. So, and before that, I want to make sure that you understand that osmoregulation and excretion are usually linked to each other in many animals, which is why we're combining these two concepts in this particular chapter. For us humans, it is the urinary system that maintains both of these processes, okay, osmoregulation and excretion as well. A few animals, though, they are not the same. So we'll talk about that when we get to those particular animals. All right, so osmosis, like I said, always refer, oh, remember my, look at my caps, always refer to movement of water. Why I always keep repeating this over and over again? Because I have so many Bio 1406 students that are still confused about this. And then when they come to anatomy, they're like, I don't know what this is. And they keep making the same mistake. Remember, osmosis is the movement of water, not solutes, okay? Not solutes. It is talking about water. And in a sense, why is movement of water equalize the solute concentration is then the fact that when we move water to one side, we're really diluting that side out and then concentrating the other side down. I'll talk about that with a picture in a bit. All right, so osmosis usually occur when there is a membrane, usually semi-permeable membrane, that separates two fluids that are not permeable to the solutes. Because, like I said, if you have a little beaker with a membrane and some fluid, but yet this membrane has holes in them and they are permeable to your solutes on either side, well, then it doesn't matter that there's more solute in one side than the other. Then they would just cross right over so that you have the same amount of dots on either side because of the fact that this membrane has these pores or holes that allow for those solids to cross over. That is diffusion. Okay? Not osmosis, not osmosis. So osmosis only happens when there are solutes inside two fluid volume that cannot pass the semi-permeable membrane. All right, so osmolarity, which I kept referring back to in the previous slide when I was going over that, remember, always refer to the amount of solutes in a solution. So if I say some fluid has higher osmolarity than the other one, then that means that one has higher amount of solutes in that particular solution. Okay, so remember osmolarity, don't let this word scare you. It just means that it is the amount of solutes in a solution. So if we say, what is the osmolarity of human blood? Well, we can measure that, and I will give it to you here from the book. It is actually 300 units of osmolarity. Now, you don't have to memorize what in the world this unit is. It's okay. Just say 300 units. That's fine with me. Okay. But I want to put the proper units in there. That's why I still put it in there. So 300 units is what the osmolarity of human blood is. And then if we compare with the osmolarity of seawater, it is about 1,000 units of osmolarity. So in a sense, if you calculate it, seawater is almost about, a little more than 3 times more higher in osmolarity or has more solutes than human blood, which is quite a bit if you're thinking about, how much difference the concentration of solutes is between us and seawater. Okay, this is the whole reason why we can't afford to be just drinking out of the ocean. We have to drink fresh water. Okay, we'll talk about that when we get to a later slide talking about um, salt seawater and everything. All right, so if two solutions have the same osmolarity, we will say that they are isoosmotic. If the solutions with one of the solution has a higher osmolarity, we will say that it is hyperosmotic. If the solution has a lower osmolarity, then we will say that that is hypoosmotic. Make sure you remember the definitions of these words. I didn't bold underline it because it was bold and underlined in the previous slide. So water will always flow by osmosis from an area or from a hypoosmotic solution to a hyperosmotic solution. Okay? Think about this in your head once again. I know that sounds like a mouthful. So water will flow by osmosis from in a sense, slow concentration solution to a high concentration solution. Okay, that's really what that means. And we always say water always move into the area with higher concentration. So that's why that statement makes sense. So this picture right here is illustrating what is happening with osmosis. So there's this container with two, volumes of fluid that is separated by this particular membrane. And obviously, this particular membrane is not permeable to all these red circles on the two volumes of liquid. Okay. So the only thing that can happen, because water is so small, is the only thing is for water to move through this membrane in order to equalize the concentration, because that's just the law of nature. Things always move from a higher concentration to an area of lower concentration. So there, these red circles are like, oh, I can't move through, but yet I know here you have higher concentration of the red circles, and here you have less concentration of the red circle. So what would you say this in terms of osmolarity word for this side? It is hyperosmotic. Okay. How about this side? It is hypoosmotic. Okay, so we already said, excuse me for my bad handwriting here. I'm trying to write while I'm, elevating my hand. Okay, so we already said osmosis is where water always moves from a hypoosmotic solution to a hyperosmotic solution. So which side will the water go? Water will move towards the right, because it always goes towards the side with more solutes, and you can see that with the red circle and with your eyes anyway. Okay, so what you're literally doing with the water is that you are letting more water come up in this fluid, which means you're really diluting that side of the fluid volume, because we cannot move the red circles around. What we can deal with is by diluting one side and concentrating one side. Because this side, let's say, just arbitrarily have 10 units of osmolarity. This one have 2. Well, there's no way for me to equalize that by diffusion. So let's dilute this down to, I'm going to 6, okay? And then we'll concentrate this down with less fluid so that both of them have an osmolarity of 6, okay? That way both sides now have equal concentration of solutes. We didn't change the amount of solutes on either side. It's not like I put a red circle into the other side. It's not that. It is just adding water to one side, removing water from the other side, diluting one side, and concentrating down one side to give you equal water or solute concentration. Okay, and that's how osmosis work in a sense. It's not this abstract thing with rules that you have to memorize. It makes sense in when you have a drawing like this and think about the whole situation. Okay. All right. So animals can then maintain water balance in 2 ways. One is where we call them osmoconformer, or we can call them as osmoregulator. What does that mean? All right. So for animals that are osmoconformers, these are animals that are always remaining isoosmotic with their environment, which means that they don't need to change their salinity or solute concentration in their body. Their solute concentration is already the same as their environment. They always keep their body the same solute concentration as the outside. They don't change. They don't need to. So usually, of course, then this is for marine animals only. Marine in this particular book means that this is salt water, okay, or from the ocean, from seawater. That's what marine means. So when we're talking about osmoconformers, there's usually no tendency to gain or lose water because they are isoosmotic with the environment. You don't need to move any water in this case. They are in a happy situation. And so it is their internal osmolarity is the same as the external environment. So that's why it's an isoosmotic situation. So they just conform their body fluid concentrations of solutes to be the same as the seawaters. So that's why they are called osmoconformers, because they change their body, in a sense, transform or conform their body's solute concentration so that it is the same and matching as the environment. Whereas the other one is not the case. For osmoregulator, these particular animals control internal osmolarity independent of the external environment, which means we don't change our solute concentration with whatever environment we're in. It's not like I'm going to jump into the ocean and now my body has like a ton of salt in it, and now I'm in this um, isoosmotic situation with the beach water. That's not the case. That doesn't happen in us humans. We humans, along with a lot of mammals, land animals, we are osmoregulators. So we don't conform to the environment around us. We control our own internal osmolarity. So if this animal is in fresh water, which means they are in a hypoosmotic environment because fresh water don't have a lot of solutes in it. That's why they're freshwater. So if you compare any of the animal cells, any animal cells compared with freshwater, animal cells is always higher in osmolarity than the freshwater, which means the animal cell is always hyperosmotic to the freshwater. All right, so in this case, we're talking about the animal being in a hypoosmotic environment. What would happen to the water? Which way would the water move? Well, which side has more solutes, the fresh water or the animal? We just said the animal. So that's why water will move into the animal. And then because they cannot conform to their environment, all this water gain, they gotta somehow internally fix it themselves. So the way they fix it is that the animal will then discharge all that excess water they're continuously putting into their body or allowing to be diffusing into their body because there's nothing they can stop it. So, but yet they live in this particular environment, so they just gotta deal with it in another way. So the only way they can deal with it is, okay, I'm just gonna pee more in a sense, urinate more to urinate a lot of dilute. Even anything dilutes urine to get rid of that all that extra water that keeps coming into the body. Now, if an animal was put in a hyper osmotic environment, which means this is from, let's say, seawater or the ocean, then you have a lot more salt. So regardless what animal it is, you dunk them in the seawater, a lot of times seawater will have a higher concentration of solutes compared to the animal cells. There will be some exceptions. We'll talk about those later. But let's say you have less solute concentration than the salt water out there. So which way will mud water move? Which way has higher salt? The environment, right? Because this is the salt beach water. So then water will then move out of the animal. So they're, in a sense, they're dehydrating themselves in a sense, in that situation. And that will be similar to if I was to go and drink liters of seawater inside of me. Okay, because that's a very, very high salt concentration solution that I'm putting in my cells and bombarding my cells with. So, in a sense, it will end up dehydrating myself because water will move out of the animal, and then in order to deal with it, animal will better take in more water to replenish. I know you're like, how does that work? Like, they're already in seawater. How do they take in more water to replenish? They are in seawater. Well, we'll talk about that. How do those fishes or animals that live in the ocean deal with this situation? So they have specific ways to deal with this. We, on the other hand, don't have any specific way to deal with it. If I drink a whole liter of salt water, I better go drink some pure water very quickly, or else I will be in the ER very soon. So this is why, in a sense, we cannot drink seawater. It will only dehydrate us even more, and it's just gonna get us more thirsty and quicker to die. So even though if you're on the verge of dying, you see all this ocean water, that's water, let's drink it. Never drink it. I know it may seem tempting, but seawater will only make you die faster if you are in a survival mode. All right. So osmoregulatory challenges are things that animals have to face depending on where they live in. So osmoregulation is what allows animals to live in an environment that osmoconformers can't. So osmoconformers, in a sense, they're pretty restricted to where they're living in is because their body is already conform to the environment they're currently living in. If you put an osmoconformer to another spot, they're probably not going to be very happy. And, depending on how flexible their range is, they would die right away if you put an osmoconformer to a different solute concentration environment. So it depends on what kind of osmoconformers they are. So osmoconformers are more restricted to their environment, but they're usually happy where they are, is because usually wherever they are, the solute concentration doesn't change by much. I mean, most likely they live in the ocean. If they live in the ocean, the whole entire ocean would not just change in solute concentration out of the blue in a very rapid pace. So that's why they're just happy where they are. But osmoregulator, they are free to go live in different environments. Now, animals that, if they can deal with it. All right, so animals that cannot tolerate substantial changes to external osmolarity, which we have those, they're called stenohaline animals. So these are, for example, oh, I don't know what happened to the E there. I remember putting the E there. Example, you have goldfishes, sea turtles, etc. So goldfishes, it's we all know they are freshwater fish. You put the goldfish in the ocean. I'm gonna release this goldfish into the wild. You're just gonna end up killing them because once goldfish is in that high salt solution, okay, water will come out of their bodies and they will die very quickly. So don't do that. Okay, so for some animals that can survive in bigger, larger fluctuation of external osmolarity, there are animals that can do that, and they are actually able to move between environments with different osmolarity. These animals are what we call euryhaline. Urahani, meaning that they can live in two situations. Their daily life or times in their life stages in their life will always alternate between two environments, low salt and high salt. So one example of urahane osmoconformers, so here are those osmoconformers that can work, two different environments, are barnacles and mussels. And that's it. Okay, so like I said, osmoconformers are very conformed to their environment. You don't have a lot of urahane osmoconformers. Barnacles and mussels, they can live in what we call estuaries. Okay, and estuaries are where bodies of water usually are found where river meets the sea, because we have that cases, right? Where there's river, a long, long river, and then they drain into the ocean. So that drainage usually have a point where the river will meet the sea or ocean. So that's where we call estuaries. What we mean by estuaries. So there's a mixture of fresh water draining from the land and salty seawater coming in, or on the outside area, excuse me. So that's why we would find these species there that are able to, survive in some fresh water, and then they're able to switch over to sea salt water, because depending on how high the tide is, and we know mussels and barnacles, we've talked about them, especially barnacles, they're pretty sedentary, sessile animals, so they're not going to move. They're stuck on that rock for barnacles' case. Remember that we said that they are stuck on the rock like glue. So when tide goes away and the river now is further out, they're in freshwater. If tide of the ocean comes up, then they're all that part of the salt water comes up higher, then they're in salt water. So that's why they're able to, I guess, in a sense, naturally selected an adaptation for being able to be urahane osmoconformers, or else they won't survive in the environment and they will be killed off. But the fact that they have that one trait and one variation that allows them to survive in both environments, they prevailed in that area. And there's not a lot of species that can do that. All right, and there's also urahane osmoregulators, and these include striped bass and some species of salmon. Why these fishes need to be in freshwater and saltwater at the same time or in different stages of their life is because both of these fishes have to migrate up the river to spawn. So, spawn means they're laying their eggs, releasing their eggs in a particular area. So supposedly these eggs don't survive very well in salt water. So that's why these fishes have to give it all they got, all the energy they got, all their life they got to, especially for salmon. I know the story of salmon. They have to swim against the tide, the movement or the tide waves on the river, and they have to swim against it using all their energy. And by the time they get up to the top where the river is, they will spawn, release their eggs, and because they've been working so hard all that time getting up there, a lot of time, a lot of times they would die off by the time they finish spawning. But when those eggs are hatched within the fresh water up there, that time when they're little baby salmons or baby striped bass, they are living in freshwater environments. They are able to thrive in freshwater environment because they were born in the river. But of course, as they grow up and mature to be adults, they will move and swim towards the ocean where their, parents were once in a lifetime. So once they move to the ocean part of it, then they acclimate to the salt water when they live there. Okay, so this is why there's your haline osmoregulator, also an evolutionary adaptation that these fishes have overcome in order to reproduce. Because probably those fishes that can only spawn in sea salt water all have died off. There's no reproduction. Remember, the criteria for natural selection is you have to be able to survive and you have to be able to reproduce. If all of their babies are dying in the saltwater, all of their eggs are dying in the saltwater, they're not going to have another population in the next round. So that's why those that have that variation of jumping up and going towards, up in the river, they will be able to spawn and have a successful next generation and let the population keep going. So that's why nature have selected for this particular trait in striped bass and salmon, and specifically sockeye salmon. I forgot to say that. All right, so most marine invertebrates are osmoconformers, like I was talking about. So remember, osmoconformers, they have osmolarity that is the same as the seawater. They have no challenge whatsoever in water balance because whatever's inside their body, it is the same solute concentration as the water around them. So they don't need to have the problem of keep peeing a lot or pee little and get this salt out of their gills. They don't have that problem. Now, but if they need a specific solute to be at a different concentration than the ocean, then they have to use energy to actively transport those. Notice active transport, because a lot of times you're transporting these ions against the concentration gradient, and that's why you have to put in ATP and put in the work to transport those ions out. One of those examples is the Atlantic lobster, which maintain a much lower magnesium ion concentration, much lower than seawater. So for them, they like magnesium concentration at about 9 millimoles of concentration, whereas seawater has about 50 millimolar of magnesium. So that's why for them, these lobster then, even though they don't have to deal with all the all the other ion concentration and all the water concentration, they just have to at least deal with the magnesium that they want at lower concentration. That's just one example. So marine vertebrates then have evolved two osmoregulatory strategies to overcome a dehydrating environment of seawater. Now we're talking about the seawater, how those animals are able to survive in such a high salt environment is what we're going to talk about in this particular slide. So there's two ways. One way is that they will balance their water loss by drinking a lot of seawater, like literally gulping it down into their body. So, and then for example, fishes that do this are bony fishes. And remember, bony fishes, back from a previous chapter, we said these are your rayed fin fishes and lobe fin fishes. So what they do is that they know they have all this excess salt that is being ingested when they gulp all the seawater down. So those excess salts actually have to become, actually is able to be eliminated through their gills and also their kidneys. So their gills and kidneys is able to excrete salt ions, and a little bit small amount of water in their kidneys. Their gills can also do the same thing. Okay, so yes, they're gulping in all this water, but their body can, in a sense, filter out those extra salts, get rid of it through their gills, get rid of it through their kidneys, and only retain the bulk of the water that they're gulping down. So that's why here it says when they're gulping down that water, it is gain of both water and salt ions from either food that they eat or by drinking and gulping down that seawater. But they will be able to absorb as much as they can from the water by gulping it down and excreting the salt out of their bodies. Yes, water will continuously be coming out of their body. It's a much harder job here we're talking about, because whatever comes in a sense, is kind of like they come right back out, because it's a losing situation, but they win a little bit. That's why they survive. Remember you said when an animal is put in a sea salt water situation, water always move out of the animal because they go into the higher salt concentration environment. So as they're putting in water, that water is coming right back out through their skin by osmosis. So this is why they have to constantly drink a lot of that seawater and get rid of that extra salt with their gills. And of course, to conserve whatever water they have, they probably don't pee out a lot of urine. So that's why it's just a small amount of water in urine, because they can't afford to lose any more water than they already are losing to their environment. So one example of bony fishes are cod, and this is what this example is of. Okay, so second evolution or adaptation that these marine animals can overcome the salinity of their environments. Salinity means the salt solution, the salt concentration of the environment. So they have high concentration of several other solutes that is also impermeable through their skin, cell membrane, and they just retain a lot of solutes in their body, such that whatever total solute concentration that is inside their body, it's either equal to the ocean or it's a little more than the ocean. Because remember, when we're talking about solutes, we were, kind of simplifying by saying, oh, there's a separation of the two fluids. There's orange circles and red circles in there. That's just a simplified thing. In a particular animal, at one point, there's not just one type of solutes. There may be some triangles. There may be some open circles. There may be some closed circles. There may be some rectangles. Whatever it is, it's a whole mixture of solutes in the animal cell. And as long as the total amount of solute concentration is the same on both sides, then it doesn't matter. Maybe you have 5 of this one thing, 5 of the other, and then 10 of the last thing. Well, that adds up to about 20. So whatever this side have, as long as they add up to 20, you still have the same 20 units of osmolarity on either side. It is still the same concentration in an isoosmotic situation. So that's why, if that particular species of animals and fish, they have more solutes that can be retained and trapped inside their cell, that makes them more either equal to the ocean solutes or higher than the ocean solutes, then they won't be losing water. So one example of that then are sharks and other conrichthians as well. We talked about conrichthians in the previous chapter. So all of them have an ability to do that. So let's talk about sharks. So for sharks, they have a lower internal salt concentration, like other fishes have the problem of in the sea, but they have high concentration of other solutes such as urea and also trimethylamine oxide, or also, if in abbreviation format, TMAO, okay? TMAO, trimethylamine oxide. So it doesn't matter what it does, okay? So it is a type of lipid that's in, I think, is a type of lipid molecule, organic molecule inside the shark's cells. And so with that and the salt concentration they have added together, their total concentration of solutes in the shark is actually a bit higher than seawater. Just a bit, not too much, just a bit higher. So even though, the shark can ingest food that has some salt in it and a little bit of salt water in it, their gills can block loss of urea. That's their little adaptation. And then water can be absorbed by the gills and the skin because of the fact that the body, the whole entire shark's body and all their cells have slightly higher solute concentration or hyperosmotic to its surrounding. So that's why water tend to be gained into the animal. All right. So most of the urea are retained by the kidneys. So whatever little salt they have ingested then will be excreted in urine by rectal gland. So that is another thing that they have are these rectal gland that is able to get rid of any extra salt they have. So notice that's why in seawater, let's say salinity is about 35 units, and then their blood salinity for the shark, it's 35 plus, which means a little more than the surrounding around them. So because water is, the shark has a higher osmolarity, hyperosmotic to the environment, water will slowly enter the shark's body by osmosis. So that's why when you see a shark, you don't see them gulping down water. Sharks actually don't drink. They don't drink any water. They just eat. And they just eat their prey whole. That's all. So whatever water gets trapped in that prey or while they're chomping on that prey, that's the water they have to drink accidentally in a sense, along with the swallowing of the food. It's not that they're gulping water down. Water just freely move into their body by that slight osmosis. And then any excess water and salt that they have taken in from the environment or while they're eating will then be disposed of in urine, feces, and also that those salt secreting glands that I was talking about, and is located their rectum. So you will hear or you will read about people saying salt secreting glands or rectal gland as well, but they're the same thing. They're just they sense that there's more salt coming through the body, they would just excrete it out for the shark itself. So that's something very cool. They don't have to be like the other fishes where they're just gulping a lot of water constantly. They just normally have that capacity. Again, an awesome adaptation that sharks have to live in such a high salt environment. All right. And then what about for freshwater animals? What do they do? So for osmoregulatory problems of freshwater animals, then really are then the opposite of marine animals. While in sea marine animals, they were in a very high salt situation and they're afraid of losing all their water. They're afraid of dehydration. So when we're talking about freshwater fish, then they face another problem that's opposite of that. They're not worried about their environment taking water away from them. They're worried about the environment putting too much water in themselves. So because when a fish is in a freshwater environment, the animal itself is hyperosmotic to the environment, which means the animal has more salt inside. So water will then move in from the environment into the fish itself. And I have already said, when an animal is in freshwater, a lot of times their animal cells are always hyperosmotic compared to the freshwater. So that's why freshwater animals then will have the problem of gaining too much water if they don't have some sort of adaptation that gets rid of them. So, solution for freshwater fish is, I guess, in a sense, simpler than saltwater fish. They just need to then excrete large amounts of very dilute urine and drink almost no water. So, the saltwater fishes, they have to keep gulping down water, conserve water, don't pee as much. For freshwater fish, don't drink and they have to keep peeing without even drinking anything because water will just come into their body by osmosis. Now, while all that water is coming into their body cells, they can also lose a lot of salts. So what they will do to recover the ions and salts that's coming in from all that, or being diluted by all that water, they would actually eat and take up salt across their gills when they are putting, letting, I guess, gas exchange with the water environment. They would do that. They will try to, capture all the salt in their gills so that they are constantly replenishing their body with a little bit salt as they're being diluted by the water around them. So just like what this picture is saying, you have gain of water and some ions within the food that they eat. They're already not drinking, okay, but they still have some water going into their body because they gotta breathe. When we already talked about how fish breathe in the water, and then also the food that they eat, and they also gulp down some water along with it. So then the uptake of salt ions is be possible by the gills. The gills will try to trap some ions, and then water will just gain and go inside the fish freely due to osmosis. So what they would end up doing is all that water that's just constantly moving into its body, let's pee them all out. So excretion of salt ions and also large amounts of water in dilute urine from their kidneys will be their adaptation against being in the freshwater environment. So for urinating salmon that we talked about, the sockeye salmon in the river and streams, they are in freshwater environments. So they will produce a large amount of dilute urine to try to get rid of all that extra water going into their bodies. Their gills will also take up some salt, whatever salt they can get from their environment. And then once they're, done spawning, they die. But then their babies are also born in a freshwater environment. And then once they grow up, they move into the saltwater environment. What will they do? So once the salmon is in the saltwater ocean, they will produce something called cortisol. Cortisol is actually a steroid hormone that will increase the number and size of a specialized salt secreting cells. Okay, so it's just specialized salt secreting cells. Just like that, those rectal glands that sharks have, salmon also has this particular salt secreting cells that can, whatever extra salt they're getting, they will excrete it all out through these particular cells. So that's why once they are in saltwater environments, cortisol will kick in and start to tell these cells, you got to get bigger and do cell divisions more. We need more of you around to get rid of all this salt that we're getting in this new saltwater environment where water is now moving away out of their bodies, and that's a problem, dehydration problem. So of course they switch over to not peeing from peeing a lot. On top of that, they have these salt secreting cells that will help, sort of get rid of any excess salt that has come into their bodies. So that's a pretty interesting story. All right, so with that, I just want to show you a video that kind of where, the guy from Crash Course. I love Crash Course. It's like the way he talks is really a summary of the entire concept or the entire chapter in a lot of times. And I saw he has a talk on do fish drinks, and he kind of talks about how freshwater fish, and saltwater fish osmoregulate. So I just want to give you another view so you have another person explaining it in a different way. Hopefully there's no copyright issues, but if there is, I will just...
All living things on Earth need water, because the chemical reactions that make life possible happen in an aqueous solution. Which means us land animals have to drink it to keep it inside of us all the time. But what about fish? It seems like since they're always in the aqueous solution of whatever body of water they're swimming around in, they wouldn't need to actually drink the stuff. But it turns out that some fish actually do need to drink, and others do not. It depends on the fish and what kind of neighborhood it lives in. Water, you should know first of all, is really into finding a balance. If you have a membrane that water water can pass through and there are different concentrations of salt on each
That's a good
side, then the water
review.
will always flow toward the saltier side of the membrane until the concentrations on both sides are the same. That means that freshwater fish have it easy. Since the concentration of salt in their bodies is higher than the concentration in the water, that freshwater just flows right in, mainly through their gills and into their bloodstream. They also swallow some water when they eat. I mean, it's kind of unavoidable, but they don't need to actively gulp down water. But in order to retain the balance of concentrations, the fish's tissues still need to have some salt in them. And that's where chloride cells come in. These are special cells in the gills that produce large amounts of an enzyme that controls the flow of dissolved salts like sodium and potassium across cell membranes. So in freshwater fish, these chloride cells work hard to bring just the right amount of salt into the fish's bloodstream. Now saltwater fish have the opposite problem. For most of them, the concentration of salt in the water is higher than the concentration in their blood. That means that as they pass water over their gills to breathe, the higher concentration of salt outside of their body is constantly sucking water out of them. So these fish have to drink a lot, which they just gulp down by the mouthful. But since the only water around is salty, they have to filter out the salt to make it safe for them to absorb. A lot of that salt is filtered out in the fish's kidneys, but these fish have chloride cells in their gills too. It's just that in their case, those cells are constantly pumping salts out of their bloodstream and into the saltier water. So they may be surrounded by water, but no matter where they live, fish are always looking to find that balance. Thanks for asking. If you have a quick question you'd like to ask us, we're down in the comments below.
So that is pretty much kind of another view of what I talked about, how the fish in saltwater versus freshwater is able to do water balance. Oh, okay. All right, so we talked about, animals in saltwater, freshwater, hyperosmotic environment and hypoosmotic environment. What about animals that don't get any water? Now, this is a pretty interesting topic. There are animals that can actually survive desiccation. And remember, desiccation, we talked about that, is when, we're depleting an animal's body of water by a great amount. This is talking about extreme type of dehydration. It's not just not drinking water for, 1 hour or two. We're talking about extreme dehydration, literally taking 90% of your water away for something, in a sense. So, in this type of extreme dehydration or desiccation, most of the time it is fatal for most animals. But there are a few aquatic invertebrates that usually live in temporary ponds or some film of water around the soil, because that's how they usually dry out. That's why it's a temporary pond that can dry out, or films of water around the soil. They can also dry out over time depending on the season and temperature. So that's why they have this capacity to survive with very, very little water. So technically speaking, there are some of these few aquatic invertebrates that can lose almost all their body water and still survive. And these animals, they enter, of course, they even in that you're talking about, taking a lot of their water away. So they're not walking around very happy, They are just in what's called a dormant state when the environment dries up. And this is an adaptation that is called anhydrobiosis. Anhydrobiosis, an means no or none, hydro means water, biosis means life. So that's why this whole word means life without water, anhydrobiosis. And so it's not that they can't eat and start doing and walking around, running around like crazy and being happy in a dry up environment. It's just that they can survive. So, because they go into that dormant state, I guess, like I said before, it's kind of like that hibernation state that some animals do in the winter. But in the winter, just a couple months. But these guys can do it for years. So I already talked about this once before, but here it is again. So tardigrade, or what's commonly known as water bears, they are aquatic invertebrates, very, very tiny, microscopic level, less than 1 millimeter in length. They can actually go from 85% body water and then completely dehydrate to just less than 2%. And they can actually stay so for years. So we're talking about going from this nice round looking bear species walking around nicely, and they're just eating and feeding, and then when they are removed water, they're in this little tiny mush, all crumpled down, dry as death, and they can actually survive that way for decades or more. They've tested it for 10 years, and I'm sure it can do longer than that. And I think, I mean, when you look them up on YouTube, you would even see that some people said they were actually able to survive in space for a couple days. So somehow I think some of them accidentally got into a spaceship and then it got out of the spaceship. It was on the outside or something, and they were able to still become alive when they come back inside or something like that. So that's pretty crazy. So they think this is a species that can survive in space. So we don't know. That's maybe a myth. But they say when after it's been dried up for many, many years, all you have to do is just add water to rehydrate it. They will suck up that water and rehydrate within a couple hours, and then it was just start moving and eating like nothing had happened. Um, which is pretty cool and bizarre. And so to do this, an important adaptation is that these water bears have gotten in order to do this is how can they keep their cell membranes intact under such dry conditions? Because when we're losing water, your phospholipids, all that plasmic water, it's gone. So you're really crumpling down all the macromolecules that you have in a cell. Okay, all the cytoplasm, all of what's inside the nucleus, you're really smushing them down like paper. So, what is it in these water bears that's still keeping all these macromolecules in their cells alive? Because they're multicellular organisms. So studies about and hydrobiotic roundworms, because turns out that water bears is not the only invertebrates that can go through anhydrobiosis. Roundworms can also do it. A specific species of roundworms. Remember, they were from the phylum Nematoda, as we've talked about in a previous chapter. So they were show that desiccated roundworms actually contain large amounts of a disaccharide called trehalose. Okay, trehalose. They think this is a sugar that can protect cells by replacing water that normally associate with the protein and membrane lipids, and in a sense, the trehalose can stabilize them just like water can. So somehow this sugar can take the place of water and do what water normally do to keep these, organelles within the cell still alive. And then when you hydrate it, it will just come back to life. So, on the same note, they also found that many insects that can survive freezing winter can also use trehalose as a membrane protectant. Remember the last time we talked about these water bears? We said that they can survive temperatures as low as -80 Celsius, which we said, oh my goodness, that is crazy. And so that's another reason why these water bears can survive in such freezing temperatures is probably because they have trehalose. And then plants that are resistant to desiccation also have been found to have trehalose. So there's something cool about this particular sugar disaccharide that is allows it to be able to protect important macroorganelles like proteins and or even macromolecules like proteins and DNA, probably. Hence, recently, scientists learn from these observations of nature, because we always do that. We humans love to learn and observe our environments. Whatever's interesting, we take it and copy it and see what we can use it for, and sometimes even make it better. So, back in the day when we're working with DNA, protein, or cell samples in a research lab or in any lab setting, you would have to keep those samples frozen is because of the fact that they will degrade over time. But what if this trihalose can actually protect these macromolecules from degrading? Because, what we store these samples in usually is -80 degree freezers. And for my lab, when I was in a research lab in graduate school, we actually kept like really long, like old samples that we don't want to trash in -150 freezer, and they always break over the summer. So, on top of always breaking, they always use up a lot of energy. So, this is an energy waster in a sense. Now, if we can actually keep these molecules in room temperature and still be able to stabilize it, then that's a different story. Then we will be able to save a lot of energy if we don't have to use freezers to keep these molecules alive. So, they've tried to use some form of trihalose to stabilize these particular molecules, and I think they've been successful. So that's something interesting. All right, so I wanted to show you how these tardigrades are able to go into that anhydrobiosis mode and then just rehydrate themselves and be able to climb back up. So this is a pretty cool because and you can see it move too, because the whole reason why it's commonly called water bear is because they walk around like a water bear. So let me show you this video. And see, that's how they move, like a little bear crawling around with, sharp claws, but they're microscopic. And they're just looking around for food, and you will see the person now that's when they have taken away water from the water bear. The water is drying up. It's in this little tiny bit of water left, and then slowly completely dry up, scrunching and scrunching and scrunching. As it dries up, it just keep losing water from its body, losing water, and then eventually it'll become a pretty much a clamped clump of dust, like the book put it. It's a piece of dust that is just in this super dry state. You see, completely like clamped down like dust. And then they will rehydrate the animal with water and see what it does. It just takes up that water, be able to move again. I know they kind of skip more in this particular video, but on YouTube there's actually more videos like this where they are able to show more progressive stages of the water bear, soaking up the water and be able to rehydrate and walk normally after that. So we are at 1 hour and 5 minutes. Hopefully you have enjoyed this part of water bears. I thought it was a pretty interesting story, and then we'll continue in part 2.